Buenas tardes, es un gran placer presentar esta charla para ustedes. And I would like to thank the organizers for this wonderful meeting, starting with Maribel and Gabriela for the invitation. And I'd like to thank also ISCB and the very efficient team of Diana, Mallory, and Seth, and everyone who made this meeting possible. Okay. So the number of shotgun metagenomic data sets is growing. And this is a very simple graph that I put together simply by typing some keywords into a PubMed search. Usually these kinds of graphs show exponential growth, but that's not what I got when I did this. And it's most likely an undercount because it relies on what has been published. It's known that there's a lot of metagenomic data sets that are not publicly available. But in any case, the premise of this talk is that these data sets can be mined to create what I call microbial virtual surveys. And what do I mean by that? So given a bacterial genus, we can ask all these questions. How many of its known species can be found? What are their relative abundances? Is there anything interesting about environments where they can be found? Also, compare species versus abundance versus location. And of course, you can ask your question about any given genus. Are there novel species to be discovered? And I think yesterday another speaker mentioned this this piece of news here. It came from Science magazine. It's an interview with a scientist who says that microbial should not be considered second-class citizens. They also deserve to be recognized for their value and preserved and put on the list of threatened species. And Cuatro Ciénegas was mentioned in this article. And it's evident that in order to protect microbes, you need to know that they exist. And this is part of the motivation of what I'm going to show you. These surveys could be done with existing tools. I want to be very clear from the outset about that. So I list here a few: Kraken2, Kaiju, MMSEX2. But these tools are optimized for speed and or memory usage for very large data sets. They're excellent for a general profile of the microbes that you have in any given data set. But another premise that I have is that a user will provide me with a target genus. And therefore, I asked myself the question, well, if I know what the genus of interest is, what can I do to improve on these other tools which are general purpose? And the hope is that having a target genus, you will have more precision at deeper taxonomic levels, specifically species and strains. And so we developed this tool that provisionally we call the BH tool. And it's not very complex, as you will see. It's based on BLAST, though the similarity engine is simply BLAST. But we put a lot of effort in this reference data set that we call GRS, the Genome Reference Set. So let me explain to you what that is. We built one GRS for each target genus. And it's more or less manually built. And which means that it's basically a curated genome set. You may know that NCBI has a problem of mislabeled sequences, and so I didn't want to go ahead with genomes from NCBI, relying 100% on their labels. And the curation we do is kind of simple. We only use isolate genomes and genomes that have at most 100 contigs. So at the first approximation, we rely on the labels for these ones. But later processing, if something comes up that is an outlier, we will be able to identify that as a mislabeling. And in order to build this genome reference set, we use a mathematical model called maximal independent set. It's a well-known problem in graph theory, and I'll explain to you very briefly. So here you have 5 nodes. Each node represents a genome, and they are all connected because we compare all against all. And the blue edges connect 2 genomes that have less than 90% average nucleotide identity. And the red edges connect vertices or nodes that have more than at least 98% average nucleotide identity. Okay, what we want is to extract from this graph a set of representative nodes, which will allow more efficient processing downstream. And for that, we use the maximal independent set problem. And it's relatively simple to explain. So you remove all the blue edges, and you keep the red edges. And after you keep the red edges, you try to find a maximal set of vertices such that there are no 2 nodes being connected by a red edge. And so in this particular case, we would end up with 4 nodes, and you would throw away one of those, the one marked with the red X. And we, to choose between these 2, we choose arbitrarily. Okay, what does this gain us? So it's more economical than using all NCBI genomes for a given genus. And as an example, one of the results I will present for you is for Acinetobacter. And in Acinetobacter, in NCBI, when it did this a few months back, there were 14,337 genomes. And with the GRS, we got a reduction from 14,000 to 1,000. So it's a 14-to-1 reduction. But perhaps more important than that, or equally important, is that it gives us more resolution at the species level. And that greater resolution is part of the novelty of what we are developing, as I'm going to show you. It's important to say that even though the concept of a maximal independent set is relatively simple, we're dealing with graphs with thousands of nodes and millions of edges, and we need an algorithm to find the maximal independent set. And the algorithm that we use was developed by Luby, and it's a classic computer science distributed computing algorithm. Oh, thank you. So I keep talking about strains, but strains, I'm not a microbiologist by any means, and strain for me is a strange concept from a computer processing point of view. I keep seeing names such as strains, lineages, subspecies, genovars, and other names. And all of that is very confusing. And instead of using any of these biological concepts, we use the concept of subgroups. For a given species, a subgroup is a genome for that species that is represented in the genome reference set that I just explained to you, right? So in that graph that I showed you, that toy graph, it had 4 nodes. Each of those 4 nodes would be one subgroup. So a given species may have just one subgroup, or it may have several subgroups. And you could kind of say that there may be a relationship between the subgroups of a given species and the concept of strain. But we don't make any claims with respect to that. We simply say that the subgroup is an operational concept in the context of this tool that I'm going to describe to you. So this is the pipeline that describes how the tool works. I'm not going to explain that to you, but it's important to mention that the input is context. So we assume that whatever data set we are working on has been assembled, and we are dealing with the context. My student, who's the main developer here, thinks that we should extend this to at least 2 long reads, and we are in the process of debating it. And I may well agree with him. And a subgroup assignment, if a contig has at least 98% identity to a GRS member, then it receives its species and subgroup labels. Now I'm going to show you results of applying this tool to a data set, a large-scale genomic data set. And the data set that I will use is the metasub data set. So I'll spend a few minutes explaining to you what is metasub. So metasub stands for metagenomics and meta-design of subways in urban biomes. And it's the brainchild of Chris Mason from Cornell University in New York. It's a global network of scientists and clinicians developing knowledge of urban microbiomes by studying mass transit systems, the built environment, and hospitals. They published a paper in Cell 4 years ago with this title, A Global Metagenomic Map of Urban Microbiomes and Antimicrobial Resistance. And this is a figure from that paper. Please ignore the edges that flow from these red dots from one to the other. I just wanted to show you the extent of the sampling that they did. And so these red dots represent the cities where they sampled. And so Brazil is represented here by São Paulo and Rio de Janeiro. And surprisingly, Mexico was not part of this sampling. But the Metasub Consortium keeps doing more and more sampling. And this was back in 2019, 2020. And it may well be that Mexico now is a part of the consortium. I just don't know. Okay. So what did they do? They do in subways across the world, surface swabbing from high contact surfaces, including ticket kiosks, turnstiles, railings, seats, or benches. All sequencing done at the Hudson. It was all Illumina in the Hudson Alpha Genome Sequencing Center, and with some control for contamination. And I want to go into that. So here are the basic data for this data set, 60 cities, almost 5,000 samples, and the contigs, which is what we were interested in, about 80 million contigs, with a minimum size of 500 base pairs, a maximum size of 3.5 million base pairs, a mean of 1.2 base pairs, 1.2 kilobase pairs, which means that the vast majority of this context is under 10 kb. And for the results that follow, we used the minimum as 2,000 base pairs because we wanted to be on the safe side, even though we were careful about all the classifications that we're doing. It's a known fact that the larger the sequence you have, the more reliable the classification is. Okay, so I will show you results for 3 genera. 5 minutes? I thought I had 45.
Sorry.
Okay.
You have
Thank you. So I'm going to show you results for 3 genera. One of them is Acinetobacter, as I said previously. The genome reference set has 90 species. And in terms of contig classified, we were able to classify almost 520,000 contigs with at least 98% similarity with a member of the GRS, whereas almost 1 million contigs had to go through the bottom part of the pipeline that I showed you for additional processing. Which means that this 98% that you see here is not really crucial. You might ask, well, what if it's 97% or 99%? So the method is relatively robust to this number. Okay, so we detected 85 species out of 90, and here's the graph that shows the different species that we found. And as I said, the subgroups, I think they are a powerful tool for understanding the distribution of these various sequences at a finer resolution than it's allowed by existing tools. So I'll give you a few examples. One is Acinetobacter radio-resistance. It was 5% relative balance among contigs assigned to Acinetobacter. And it's species with just one subgroup. But there were species with more than one subgroup. One of them was Acinetobacter baumannii, and you may know that Acinetobacter baumannii is part of the escape group that Gabriela was talking about. It's a bacterium that is creating havoc in many hospitals, and so we detected 23 subgroups, and one of them is dominant with 95.5% of contigs among those classified as Acinetobacter baumannii. Another one is Acinetobacter pti. It has 114 subgroups out of 119, which means that we detected 114. And again, there's one which is dominant with nearly 66%. And one thing that we have just started to do is to correlate these classifications with the metadata from the metasub data set, right? They have, for every sample, comes from a given place, generally from a given city. A given city will have several samples from different places, and the resolution that I'm going to show is by city, not by sample. And so this is a presence-absence graph of the Acinetobacter species, which are in the rows, and in the columns you can't read that, but these are the different cities. And the point is that this distribution is not uniform, right? It clearly has some sort of structure. These species over here are clearly present in the cities to the right, and for the most part absent on the cities to the left. And this is a heat map where we take into consideration the abundance, relative abundance. You can't read, but I made a zoom, and in the zoom, you can see that, again, there is some structure here. There's non-uniformity. And so for this Acinetobacter here, which is called Yersinia, it seems to be particularly abundant in this set of 5 or 6 cities over there. We tried to correlate, do a correlation analysis to see if we could understand the pattern, but there is, of course, structure in this, but in terms of geography, we couldn't see any explanation for these clusters. In terms of subgroups, So this is the same graph that I showed you before, but instead of species, now on the rows you have the different subgroups. Again, it's there is structure here with presence on the left, mostly presence on the left and absence on the right. And this is the heat map. And this is a zoom of the heat map, showing again that something seems to be going on with these cities with respect to this subgroup of Acinetobacter ursinii. Okay, so that was for Acinetobacter, and I want to show you at least one other genus, Xanthomonas. The genome reference set has 38 species. This is going to be a repeat of what I just showed you, just changing the genus. And all species were detected in this case, which was a surprising result, because Xanthomonas is well known for being a plant pathogen. It's found in many plants all over the world, but it was kind of strange to see it pop up in subways. But that's what the data is telling us. There was a clear predominance of one species of Xanthomonas campestris, and much smaller fractions for the other ones. And again, we had subgroups. For campestris, we had 36 subgroups, one of them dominant with 85%. And this other species, just one subgroup, similar to what we saw for Acinetobacter. And again, this is the heat map with taking into account relative abundance for the species. And again, something seems to be going on here at the top. This is the Bundabergiensis. This is a zoom of the previous picture. And the third and final genus is Stutzerimonas. This genus previously was under the genus of Pseudomonas, but Pseudomonas, you may know that it's a very diverse genus, and apparently the taxonomists decided that it should break it up, and at least this Stutzerimonas was a result of this breakup. The genome reference set had 14 species. All species were detected, and the distribution is, again, this with Stutzerimonas stutzerii, which happens to be a nitrogen fixer, being the most prevalent one. And again, subgroups, we detected 30 subgroups out of 34, and one of them is dominant with 86.1%. And Stutzerimonas balearica was just one subgroup. So the results, they have the same flavor. We change the genus and we see things behaving more or less in the same way. Of course, we chose these three genera because we knew beforehand, thanks to that Cell paper from 2021, that these genera were present in the data set in abundant numbers. So that's one of the reasons why we chose these three genera. Okay, so here I just want to address a little closer this question about the metadata and the subgroup. In this case, the subgroup whose identifier is 0 for Stutzerimonas stutzerii. So you can see that it's highly abundant in this group of cities. And the group of cities is listed here, right? And what is baffling us is that we look at this list and we don't see any meaning at all. Why would Rio de Janeiro and London and Sendai and Doha share this particular feature? You could argue that, well, you know, subways are a place where everybody mixes, and there's the global travel industry, and so in any one city you would see just a mixture of what's going on all over the world. But if that were the case, we wouldn't see structure in these heat maps. And these heat maps definitely have this non-uniformity. But for now, it's a mystery why that is, right? It could even be that this is an artifact. I hope not, but we are still exploring that. Okay, I'm now nearing the end of the talk, and it turns out that we could apply the same tool to another data set. And this other data set is for fermented food. And it was published this year in iScience. The corresponding author is Paul Cotter, and Nicola Segata is one of the authors here. And it's a genomics-based investigation of acetic acid bacteria across a global fermented food metagenomics data set. And I happen to know Nicola Segata, and when I saw this publication, I wrote to him and I said, hey, can you send me, can I have access to this data set? Because it's shotgun data set, right? And he said, fine, no problem. And so the data set, I'm going to describe it to you. More than 3,000 samples, 47 countries, a number of contexts not unlike the one for MetaSub. If you remember, MetaSub had 80 million contexts, and in this case we have almost 90 million contexts. Minimum size 1,000 base pairs, maximum size 2 million, mean is 2.3 kb, so the vast majority is again less than 10 kb. And I will show you results just for Acinetobacter, and I won't go through all the tables that I've shown you for the MetaSub dataset. Because here, I just want to make one simple comparison between the two datasets. And so, this is the distribution of different species of Acinetobacter for the fermented foods dataset, and this is the distribution of Acinetobacter species for the MetaSub dataset. The colors don't match, and because of that, instead of looking at these graphs, I suggest you look at the following table. So the table shows the Acinetobacter subgroups that we were able to find in both data sets. So here you have Acinetobacter baumannii, the subgroup ID is 0, and among all contexts classified as Acinetobacter, this subgroup has 95%. So it's one of the dominant subgroups. And in the global foods, it has a similar number. And for PTI, we have 65 and 32, so one is half of the other. And then Schindleri is 10% and 10%, similar, but Schindleri, there are two subgroups. And Wolffii has 4% here and 15% there. So what does this mean? I like to think, and I'll be happy if I hear comments to the contrary, that there should be some connection between what you see when you sample subways and what you see when you sample fermented foods across the world. Not a particularly good connection between the two, but they shouldn't be completely dissimilar. And the point here is that this table shows that, yes, there seems to be some connection. Okay, so that's what I wanted to present to you. In summary, the approach that I explained allows a data set specific census of specific bacterial genera. In principle, could also be applied to several data sets. And the more representative these data sets are of the biosphere, the closer you would be in doing a global census for that particular genus. And my long-term goal is to get there. But of course, in order to do that, you would need a big enough computer, which is currently not my case. But I want to stress that everything that I showed you is, for now, is just a proof of concept. I think there are still kinks that need to be fixed, and before we can start making observations and collecting results that really make sense and allow for a better understanding of distribution of a given bacterial genus across the world. But I hope that in this audience, there are people who have access to metagenomic data sets who might be interested in applying this tool to see what we can find in terms of this fine-grained classifications at the species and at the subgroup level. Okay, most of the work was done by my student Artur Solano. Carlos Piropo is a technician in my lab who makes sure that all computers keep running. Of course, Chris Mason is the leader of the MetaSub Consortium, and Nicola Segata from the University of Trento, as I explained, was very kind in allowing me to have access to the fermented foods data set. And this has been funded by various agencies. And with that, I would like to thank you for your attention. I'll be happy to take any questions.
Thank you very much. João, questions?
Hi. How dependent are your results and conclusions on the value of 98 that you started from? You did motivate that 98. Did you do some sort of sensitivity analysis of the results?
In terms of, in let's say the literature of this field, you will see that many papers use the 95% threshold. And 95 is a little too liberal. And so we opted for the 98% as to be more stringent because we didn't want to, we wanted to be conservative. So that much I can say to you. And as I said, the pipeline is not particularly dependent on the 98%. There is a dependency in the assignment. I said when a given contig comes along and we compare it to the genomes in the genome reference set, 98% is the threshold. But if it's under 98%, we still make an effort to try to classify that through another route, which means that it's not that it's a hard number that will divide what is kept from what's not kept. And so we didn't do any sensitivity analysis, and it's a continued refinement process, right? I personally would like to get rid of any thresholds. I like to structure the tool in such a way that we didn't have to rely on particular numbers, but we haven't reached that point yet.
Thank you, João. So, okay, just to make sure I understand. So you like using these contigs to do binning and to map it to a full genome, so that you have sort of like a reference genome at the end.
No, the contigs come from the data set. We don't do anything with the contigs in terms of assembling, reassembling, binning.
Taking fragments and assigning, that. Okay,
Yes.
So then I cannot have an idea like what the core size for a pangenome or whatever. It's the size of the piece of the.
So let me just correct something what I just said. If a contig has at least 98% to a member of the GRS, then it receives that classification and we're done with it. But if it has less than 98%, we have a process, and in that process we use binning because, as I mentioned at the beginning, one of our goals is try to identify new species for a given genus. Okay. And for that, we have this whole process, which includes a phylogenetic analysis part that I didn't mention here. But so far, we haven't been able to find any new species that we can really vouch for. But there's no mapping. So you said something about mapping.
Yes.
Yes, but you did mention something about pangenome.
Yes.
And this is something that we are planning to do.
And just because it's so interesting to have this, the food and the subways, to see what is the core or the proteins or what
Yes.
they have to do with each other.
There's a whole bunch of things that can be done. So I didn't say anything about gene content, for example. And of course, it's a question that pops immediately to mind. So if you have those subgroups that are specific to a certain city, can you compare the gene content of those two others that are not present in that city and say something about that? So there are many research lines that can have used this as a starting point. So just the beginning.
Okay. Thank you.
For the global fermented foods data set, do you have in your metadata where those foods are from?
Yes.
So does it correlate? Or how many of those cities overlap with the cities you have in this?
Yeah, they have it by country. They don't have it by city, which makes it harder to correlate. But
do you see some rough correlation?
I finished my talk this morning. We didn't have time to work on that, but it's in our plans.
Cool, that'd be cool.
Can this method, could this method be extended to discover new genus? Like, if I only know that, okay, my data set, I know the phyla, but I don't know the genus, can this be extended so that I could detect new?
Yeah, that's an excellent question, Maribel. Another apparently arbitrary decision here was to base the whole thing on target genera, right? Why not target family? Why not target class, and so on and so forth? And it's just for practical reasons, right? We wanted to go above species, and the next level is genus, so let's stop there. But in principle, there's one other thing. I have been working with genomic sequences for a long time now, and I think I have a good handle of what is, in terms of genomic terms, what is a species, what is a genus. I feel comfortable with that. Once you go up to family, things start to become a lot murkier in terms of thresholds and things like that. And so I think we need to really understand everything that's going on at the genus level, processing as many data sets as you can before we can start doing what you're suggesting.
Yes.
Well, I think it's going to be extremely useful. And is it available or is it going to
We're planning to make it available in the near future, because of course it has to be tied to a publication. So we are working on it.
Well, thank you.
Last question.
Could you please repeat how did you construct the network?
For the maximum independent set, the genome reference set, or
No, the network that.
the correlation?
Yes.
Oh, Spearman correlation, abundance, relative abundance values, nothing fancy, nothing out of the ordinary. And I can give you the details later on, but I don't have them on the top of my head.
Thank you.
We may have time for a small little question. Last one.
Sorry, I didn't want to hog it. How does your GRS differ from RefSeq at NCBI?
So RefSeq is
And I don't make RefSeq, so you can trash it.
okay. No, RefSeq is a good resource, definitely a good resource. And RefSeq, my understanding of RefSeq is that the NCBI staff, for a given species, they decide that there's one genome for that species that they will designate as the reference sequence for that species. And theoretically, it go through some special treatment or pipelines, processing, and so on, and then it gets this label. It's a ref-seq sequence. But they don't make any claims as to how related one genome is to another. All they care, as far as I can tell, is the labels, the taxonomic labels.
Okay.
They don't.
So you're using like a quality cutoff and including everything that meets that quality cutoff, whereas they're just like, oh, do you have this species name? We're gonna pick one.
Yes. Yes.
Yeah, I see
But
the advantage.
in our case, it's quantitative.
Yeah.
not qualitative.
That's great.